Hello everyone, this is D.Raja Sudhakar, serving as Assistant Professor in the Department of Bioinformatics, Bishop Weaver College, Trichy. Today we are going to see the course Proteins Structure and Functions. So this is a Unit 1 content. It comprises of building blocks, hierarchical representation of proteins, primary, secondary, tertiary and quaternary structure classification of proteins and structural databases. So anyone can identify the building. This is called Burj Khalifa. So this is the world tallest building called Burj Khalifa. And so there are different types of buildings. You can see, okay, there are different types of buildings, but the base is it has only the sand, cement, water, and the bricks and all. Okay. So this is the for the building blocks. Basic materials for a building construction is bricks, cement, iron rods, wood, etc. And apart from that, we need water. Okay. Similarly, for the living thing, especially for a multicellular organism, okay, so we need amino acids, nucleotides, bonds, and skeleton. Okay. So this is the single image representation of all the amino acids, the essential amino acids, 20 amino acids. Okay. So here we are able to see the amino acids with their physicochemical properties. Okay. For example, there are aliphatic amino acids, isoleucine, leucine and valine, and there are like sulfur containing amino acids you can see, which is in yellow color, that is methionine and cysteine, and there are polar residues. Polar residues are nothing but this whole green circle you can see. Okay, it has cysteine, tyrosine, tryptophan, histidine, lysine, arginine. Okay, so there are n number of amino acids are there. So here, like, what are polar amino acids? Polar amino acids are nothing but water-loving amino acids or hydrophilic amino acids. Okay, so those amino acids are always present on the surface of the protein. Okay, those amino acids are always present on the surface of the protein, and non-polar amino acids means they are water-hating. That means they are lipophilic or hydrophobic amino acids. Okay, so they get buried inside the protein structures. Okay, they won't be available on the surface of the protein. Okay, so these are some of the properties I am saying. Similarly, there are charged amino acid. Okay, there are acidic amino acids are there, hydroxylic amino acids are there, aliphatic are there, okay, aromatic are there. Okay, so it varies. So there are here the image represents the 3-letter code and the single letter code for each and every amino acid. Okay, so in the 26 alphabets, here we can see only 20 alphabets. Okay, so we are missing, there are 6 alphabets, nothing but B, O, X, box, and J, U, Z. Okay, juice. So box, juice. So if you just remember as a box, juice. So apart from these 6 letters, we have all the 20 amino acids are there here. Okay, it starts from alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, cysteine, threonine, tryptophan and tyrosine. Okay, so each and every amino acid has its own property and it defines its own structures. Okay, for example, glycine doesn't have any side chains, so it will be like a flexible one which is always involved in the domain movement. Okay, and there is an amino acid called proline which has a bond with its own R group. Okay, so that means when there is a appearance of proline, so there are more chances to have helical structures. Okay, so in building blocks, so this is the basic structure of an amino acid. Here you can see N, C alpha, C, okay, double bond O. So this is the basic one. Okay, apart from that we have a side chain C beta and there is a carboxyl group. Okay, so this is the side chain, the C beta is a side chain and this is the backbone, this is the main line, N, C alpha, C double bond O. Okay, it starts from the N, okay, and it ends in C double bond O. So roughly there are 300 types of amino acids are available in nature. Only 20 amino acids serve as building blocks of proteins for us. Okay, right now they are adding 2 more amino acids. One is called selenocysteine and pyrrolysine. Okay, So these are the 2 amino acids are getting added up to the standard amino acids. Okay. So what are peptide bonds? Okay, where the peptide bonds will occur? Okay, the peptide bonds will occur between 2 amino acids. Okay, you can see here this is one amino acid and here this is the another amino acid. So the OH group from the amino acid 1 and the H hydrogen group from the amino acid 2 will form a water molecule and it will cleaved off. So the rest will form a hydrogen bond, will form a peptide bond. Here you can see the green color, the shaded one, the green color is the peptide bond. Okay. So it's called a dipeptide. We say because 2 amino acids are there, so we say dipeptide, dipeptides. Okay. So hierarchical representation of proteins, basically there are 4 types we said, like primary, secondary, tertiary and quaternary we said. Okay, so the primary is nothing but the amino acids which are connected with the polypeptide chains. Okay, which are connected with the polypeptide chains, that is peptide bonds. Then it goes for the secondary structures. One is alpha beta sheet and alpha helix. Okay, then it goes for the tertiary structure. It forms a proper structure. Then it comes under next one is the quaternary structure, is the combination of tertiary structures. Okay, you can see here. So this is the one monomer. This monomer connected with the 3 monomers will form a one proper structure. Okay, best example is hemoglobin. Here you can see this is the hemoglobin structure. So it contains 4 tertiary structures. Okay, so this is the hemoglobin structure. So this is the primary structure of amino acid. The next one is the secondary structure. Okay, then you can have the tertiary structures, then quaternary structures. Okay, in general, again I am saying that is in colorful image. This is a plain image. Here you can see it very clearly. So methionine, isoleucine, lysine, alanine, asparagine, isoleucine. So this is the amino acid sequence. The lines are nothing but the peptide bonds. Okay, these are the polypeptide chains which is connected with the amino acids. So this is the primary structure. Then the secondary structure, it is alpha helix. This is the alpha helix and the beta strand. This is the beta strand. Then 3D structures with the atomic details. You can see it has the proper atomic details are there. There is a tertiary structure. Then quaternary structure. This is the assembled subunits. Okay. Then in secondary structures, okay, it has parallel and anti-parallel beta sheets. Okay, what is called parallel, what is called anti-parallel? We should know this difference actually. Here you can see. So these amino acids are connected with the hydrogen bond also in between. Okay, so when you have the pattern, you can see one structure moves on this side, this angle. Here you can see the arrow mark, and another sheet moves on this angle, opposite angle. It means it's called anti-parallel. If both the angles are moving in same direction, it's called parallel sheet. Okay, you can see the arrow marks are there. Okay, here you can see. So here is the angle is from the left to right. Here is the left, right to left. So this is called anti-parallel. Both here, both of them are moving from left to right. That's called parallel beta sheets. Okay. Structure classification of proteins. Okay, there are fibrous proteins and globular proteins. You might have here studied about it. And apart from that, there is one more thing called the membrane proteins are there. Okay, so the fibrous proteins are nothing but polypeptide chains connected with the polypeptide chains arranged in long strands, static molecules, and play important structure roles in the anatomy and physiology of vertebrates. Okay, because fibrous proteins are the important proteins which forms a primary muscle in our body. Okay, like if you go to gym, you do workout, right? When you go for workout, so your muscle will get tear off actually. So once the muscle is getting tear off, you will take some protein shakes, right? So that it contains only fibrous proteins actually. So those fibrous proteins will form the primary muscle in the primary muscle or primary muscle for the in the tarred area. Okay, so then your muscle will start to bulge actually. Okay, this is the theory behind that. Okay, that's what they mentioned. Static muscles, static molecules, and play important structural roles in the anatomy and physiology of vertebrates. Provide external protection, support, and shape. Okay, this fibrous proteins provides external protection, support, and shape. It is insoluble in water. Build upon a single repetitive structure assembled in cables or threads. Okay, best example is alpha keratin, hair and collagen, skin, bone, and teeth. These are the fibrous proteins. Okay, Then next one is the globular protein. In globular protein, there are polypeptide chains folded in spherical or globular shape. Okay, so in globular proteins there are 4 types. One is all alpha, another one is all beta, another one is alpha plus beta, another one is the last one is alpha or beta. Okay, here you can see the structures, right, clearly. So this is the all alpha protein. Okay, here you can see the full structure. It has only the helix. Okay. Okay, it has only the helix. It doesn't have any sheets, much sheets. Okay, you can see the alpha helix structure is more than 40%, and the beta sheets are less than 5%. Okay, this is the alpha, all alpha. The next one is the all beta. You can see alpha helix are very less than 5%, and all beta you can see it's more than 40%. Okay. Then alpha plus beta, so both will share in almost similar, like 50-50%. You can see alpha helix are less than or greater than 15%, and the beta sheets are greater than 10%. So it's anti-parallel, they say. Okay, you can see here. The next one is alpha or beta. Alpha is greater than 15%, and the beta is also greater than 10%. This is parallel. Okay. So next, the last one is the membrane proteins. Okay, the membrane proteins are nothing but in genomes. There are 30% of the proteins are the membrane proteins. Okay, they are the very important proteins. Are nothing but they are the very important proteins in our body. They can be the targets for the drug design. Okay, because these proteins are always present on the surface of the cell. Okay, so they are involved in the activation and deactivation of any molecule or any cell. Okay, for example, EGFR, G protein coupled receptors. Okay, like cell signaling proteins or ion transportation proteins. So these proteins are always on the surface of the membrane, cell membrane. Okay, so they will involve in the activation and deactivation of the cell. That means you can say the regulation of the cell activity. Okay, regulation of the cell activity. Okay, so they can be used as a targets for drug design. They are involved in cell signaling. Okay, so they can communicate with the receptors. Okay, then ion transportation. Okay, they involve in ion transportation, and there are 2 types in this. One is alpha. There are 2 types in it. One is helical proteins, another one is beta barrel proteins. Okay, this is the helical proteins. You can see this is a membrane protein. Okay, we say transmembrane protein or else. Okay, so this is the beta barrel proteins. Okay, you can see here. Okay, how the membrane will work. Okay, here you have the hydrophobic tail. Okay, this is the hydrophobic tail. This is the hydrophobic. This is the lipophilic. This is the hydrophilic head. This is the hydrophobic tail. Okay, so that means this is a polar head. Okay, so the ions will transfer through this one. This is the membrane. Okay, so thank you. This is the overview of the unit 1. Okay, in next class we see about the overview of the unit 2. Thank you.